Experiment 13, Volumetric Analysis, Acid-Based Titration. We'll be doing a technique in this experiment called titration. Titration is a measurement of the volume of a standard solution required to completely react with a measured volume or mass of substance being analyzed. That substance being analyzed is also referred to as the analyte. There's a couple of words in this definition that we want to go into a little more detail, one being completely. If an analyte does not completely react, you cannot accurately determine the amount that was present, which is the whole point of the experiment. This is a quantitative experiment. We want to know how much is there. Also, if we talk about the analyte being a solid, we also need it to dissolve completely. If it doesn't dissolve completely, then it cannot react completely, which also will give you inaccurate results as well. Another important word in this definition we want to look at is standard solution. A standard solution is a solution containing a known concentration of solute. Typically, it's a strong acid or a strong base. The concentration of a standard solution is determined by a process called standardization. Standardization is a process that we determine the concentration of solution by titrating it with a carefully measured quantity of a primary standard. So the question is, what is a primary standard? It is a highly purified compound that serves as a reference material in all volumetric and mass trigonometric methods. Basically, for something to be a primary standard, it has to be of high purity. It must be stable toward the air and also stick it in the oven where you're trying to get out the moisture. Absence of hydrated waters, has to be low cost, soluble in the solvent we're talking about, and have a large molar mass. For something to be primary standard, it has to be something that I feel confident that when I put it in the oven and dry it, basically when I weigh 1 gram on the balance, I have 1 gram of that substance. Pure substance. The reason that we need a primary standard in the standardization process is that we need something that we're confident of the concentration of. That way, when we do the standardization process, we're confident of the concentration of that solution that we're standardizing. Now, there's not many primary standards that fit that category of high purity, stable, etc. And there's some useful reagents that do not fit primary standard conditions. Examples: HCl, sodium hydroxide, silver nitrate. These are reagents that we use many times in titration processes, but are not primary standards. Sodium hydroxide is one we use today. The problem with sodium hydroxide is that it's very hygroscopic, which means when I'm weighing that on a balance, if I'm trying to weigh out 1 gram of sodium hydroxide, it's absorbing moisture from the air immediately. So when I weigh about 1 gram, I have sodium hydroxide as well as water in that sample. You can see that sodium hydroxide actually get wet as you put it on the balance because it's so hygroscopic. So we have to find a way to standardize that. I need to know how much sodium hydroxide is actually in the sample. Because it's not going to be that 1 gram, it's going to have moisture in it as well. So we do the standardization process where we go against some primary standard and we standardize that sodium hydroxide and then we know the concentration of it. Now, another way we can do it is like we're going to do in this experiment. We already have a standard solution. What we have is HCl as a standard solution, already standardized, value placed on the bottom. And we do a standardization against the sodium hydroxide to find its concentration. So we're doing it against another standard solution instead of using a primary standard. When we do that, it's referred to as a secondary standard solution, a titrant that is standardized against a standard solution. This is the process that we're going to do today. We're going to take the sodium hydroxide and standardize it against a standard solution of acid. And then we have our secondary standard solution of sodium hydroxide, the concentration of it. How do we know that we've completed that standardization process. Well, we add indicators to the analyte. Okay? And indicators are added to the analyte solution in order to give an observable physical change, basically showing us where that endpoint is at or near the equivalence point. Typically, there's an appearance or disappearance of colors, some changing color, precipitate form. There's many different types of indicators. Now, our goal in the titration is to reach something called the equivalence point. The equivalence point is reached when the amount of added titrant is chemically equivalent to the amount of analyte in sample. That is the point that we're trying to find. That's where the number of moles or equivalents of your analyte equals the number of moles or equivalents of the titrant, okay, on a stoichiometry basis there. We reach that, we've reached the equivalence point, and that's what we're trying to determine. However, when that occurs, you really can't tell it has happened. So we add indicators in there to show that this equivalence point has happened, okay? But the problem is you're really getting it a little bit past the equivalence point, okay? So when we see that color change from the indicator in our reaction with the excess titrant, okay, because all the analyte has been consumed, what we really see is something called the endpoint. Okay? This is your physical change that can be observed associated with the conditions of that equivalence. So if we're really visually detecting the endpoint and we're not getting what we want, which is called the equivalence point, then there must be some error in your analysis, which is true, and that is referred to as the titration error. That is the difference in volume or mass between the equivalence point and that endpoint. Well, if there's an error associated with your experiment, are there ways to get around that error? Yes, there are. One is like we're doing in this experiment. Our acid and base is the same in our standardization process as well as it is in our experimental process. So therefore, any differences is being canceled out. Okay, same difference in both experiments. Other ways is that you can do a blank, meaning no analyte, run the experiment and see how much volume it takes to get to that color change, and then subtract that from your volume in your experiment and get your value. Or you can do a back titration, which means once you get the color change, you then take the analyte and you add analyte to it to see how much volume it takes to get back to colorless, and then account that into your calculation as well. Your goal when you're doing a titration is to get that color change to be like one drop past the equivalence point. So you want to get a very faint color that's persistent for 30 or 60 seconds after you add that one drop to the analyte. And you want to get that faint color. You don't want to have a lot. You don't go past it too far. You want to just see where that one drop difference is, where we change colors from colorless to some color if you use an indicator that changes the color. Okay, first thing you're going to have to do is clean your burette. Typically, you take the burette and you add your titrant to it. That's the substance that you're placing in the burette. It's called the titrant. And you put a couple of milliliters and you basically just kind of stir it around, right? Turning the glass around and getting it to wet all the surfaces of that burette. And then you release that and put into some waste container. And it only takes like a milliliter or two to wet. Then you basically fill up your tube with your titrant, okay, making sure that you go past the 0 mark on your burette. The marks on the burette go from the 0 at the top and goes down to 50 milliliters if it's a 50 milliliter burette. And then you're going to notice this space between that and the tip. Okay, so you only can use and measure the amount of titrant added between that 0 and that 50. You can't measure anything below that 50 because there's no calibrated marks. You fill it up totally, then you open the stopcock and let that solution, the titrant, fill up the stopcock as well. Okay, you need that entire burette filled. It doesn't have to be stopped at 0. It can stop it anywhere and record the number. Okay, you just got to make sure you have enough volume in that burette to reach that equivalence point. Okay, so that's important and crucial because you don't want to have to refill the burette that instills more error every time you fill the burette and measure initial and the final reading. When you fill up that burette, you want to make sure that you have the entire stopcock filled with solution at the bottom there. Okay, you don't want to have any air bubbles. If you can see on this screen, this large bubble looking thing, that is an air bubble. So if that goes and you add that to your analyte during your experiment, then you basically haven't calibrated putting in solution there, you've calibrated putting in air, which is going to mean you added less solution than what you're going to read off the burette. So you got to make sure that you have all that tip filled with solution. Okay, make sure you don't have any of that little air bubble there going back and forth. You just basically got to open the stopcock and let it come through fast, or shake it a little bit, but you need to get that air bubble out of that. Otherwise it will instill some error in your analysis. To put in our analyte into our Erlenmeyer flask, okay, we're going to use a pipette. Now notice on pipettes, if it says TD on it, that means to deliver. Okay, and basically it's calibrated to deliver that amount. So if you add solution to the 0 mark here on the pipette, and then you let it all, add them all to your Erlenmeyer flask, then you will add 10 milliliters in this particular pipette. And notice that this is calibrated all the way to the end. That's the type you want to use a serological. Sometimes in your lab drawer there may be a more where it basically looks like a mini burette. You don't want to use that because then you got to stop at 10. Okay, you want to be able to just have it and add it, let it go, and add all of it to the solution. Typically, you take the tip and you touch the Erlenmeyer flask with it and count to 10. If it's just a regular pipette, if it's one that you got to blow out the tip, you're going to know that because there's going to be a frosted mark on the top of the burette. If it has a frosted mark, that means you have to blow out the tip. So that means you have to squirt out everything out that tube to get exactly the 10 milliliters of what we're looking for in this experiment. If you add in less than 10, say I wanted to add 3.2 milliliters, okay, in this particular one, you're going to basically go up to here would be to 6.8 is where you would add your solution to, and then you would let that go and blow out the tip and add exactly 3.2 milliliters. We have our pipette bulbs. Typically we put that on the top of the pipette and move the little wheel to aspirate in our solution, okay, to the mark we want, making sure that the tip stays below the surface of the liquid. Okay, as you get in your solution, if you bring it above the surface of the solution, then you're going to take in air and it's going to mess things up on that as well. Okay, so you want to make sure you keep it below the surface of the solution you're trying to aspirate into the pipette. Okay, the setup for titration processes, you have your burette. Okay, you got to have a ring stand to basically hold the burette, and you have your Erlenmeyer flask at the bottom there. Typically that Erlenmeyer flask is where we have the analyte, and in the burette is where we have what we call the titrant. Okay. So make sure that you fill up that burette all the way up to 0, somewhere around 0. Make sure that you get that air bubble out of the tip. Make sure you have solution all the way through it. You have your analyte in that Erlenmeyer flask. You also add your indicator. Okay, the indicator also goes in that Erlenmeyer flask. And then you're ready to go. Okay, then you start opening up your stopcock and let the solution go. Sometimes, where you can see that color change a little easier, we put a white piece of paper under it. Okay, so they put a white piece of paper, allow you to see that color change. Our goal here is to add our titrant until we get a color solution change. Okay, in this particular experiment, we're looking for a pink color. Now, you like to be a very faint pink color where you get that one drop difference. Okay, and it persists for like 30 to 60 seconds. In the process of adding your titrant, you got to remember that you're going to have solution as you're shaking this. You got to be stirring it by shaking the Erlenmeyer flask. Okay, you're going to have splashing of your sample going onto the sides of the flask, as well as your titrant is coming and splashing onto the sides of the flask as well. That's substance that you got to account for. So you should take your water bottle, your squirt bottle, and wash the sides of the Erlenmeyer flask periodically, okay, to wash down all the sample if you want to get a real good, accurate analysis. And you keep adding that, okay, and when you get close to the color change, where the color starts changing and hanging on a little bit longer, okay, and then disappearing, then you want to start going and add drops at a time, where you can take the stopcock and just twist it, okay, as half a turns, and add one drop at a time. And as you really get close, then what you want to do is kind of have a half a drop, open the stopcock and close it real quick, and just have a drop hanging on the tip, touch the side of the Erlenmeyer flask, then wash that down. You really want to get that one drop difference between colorless and pink. Okay, you don't want to get a real dark pink, because that you're going way past the endpoint. Okay, you want that one drop difference between the equivalence point and the endpoint. Okay, where you see that color change, dramatic color change by one drop. The better that you can do that, the more precise your data can be, then the more accurate you could be in getting your answer. Okay, you really want to work on that precision. Okay, you want to make sure that you get everything very precise in between runs. Now, how do you read the burette? Okay, the burette goes from 0 and comes down to 50, right, to 50 milliliter. Okay, so it's reading, the numbers are reading down, and it's meant to TD as well, it's to deliver. So you got to read the meniscus. Okay, you can see right here is my meniscus. So I have to read where that meniscus is at the top for my initial, and I read it at the bottom for my final, and the difference tells me how much I delivered. Now for your burette, you can see that each one of these lines has 0.01 difference between each one of these lines. So when you read a number, you read all your certain ones plus your first uncertain one. Okay, so in this case here, excuse me, 0.1, I said 0.01, 0.1. 0.1 milliliters difference between each one of these lines. And when you're recording the number, it's all your certain ones plus your first uncertain one digit. Okay, in this case, where it can be certain to the hundreds place, to the ones place, as well as to the tens place. Okay, because we can see that on each one of these marks. Okay, one of these is a tenth. So the hundredth place is where we're guessing, and that's where we're going to stop. So in this one, I'm looking at this. I can see I'm past 41. I'm past 41.1, that's where 41.1 is, and this is 41.2. So I'm somewhere between 41.1. I'm somewhere between 41.1 and 41.2. Okay? I'm somewhere between there. So I'm certain it's greater than 41.1, and I'm certain it's less than 41.2. So now I'm going to guess at that hundredths place of what that is. And looking at this one, I guess that that's about 41.18 milliliters. Now, you may have a different number for that 8. Okay, you may think it looks more like 7 or 5 or whatever. Well, that's the one you're guessing at, so it doesn't really matter. It's whatever you see. As long as you're making the same guesstimation at your initial as well as your final, reading it, things will be fine. Okay, so you basically got to go to the hundredth place when you're reading that burette. Now, to help you out reading the meniscus, sometimes what you can do is take a small white piece of paper, take a square, and fill it in with some black. Okay, so you have a black square. Then you take that piece of paper with the black square on it and you put it underneath the meniscus. You can see on this diagram here they have the black square showing. I'll draw some red on it right here to show you. And then the light reflects on that at the bottom of the meniscus, and then you can read it a little easier. So you can take a little piece of paper, draw that little square, fill it in with some black, and then put it underneath the meniscus and have the light reflect to give you make it a little easier to read the meniscus. Now, your first run is going to be, typically you don't know where the endpoint is going to be, so you go slowly to get to that first one. After that, your next experiment run, the second run, the third run is going to be should theoretically be exactly the same point. So you know where that volume should be. So if the first one took 10.5 milliliters to reach the endpoint, then what you're going to do is on the next one, add like 8 milliliters real fast. Just go all the way to 8. Then slowly creep up onto the 10 from 8 to 10. So your first one's usually slow, and the second and third one are a little faster. Okay? Same thing when you do the unknown. The first one's going to be slow, then the second and third one you should be able to do faster because you know exactly where it's going to be. So you can add that volume up to that point and continue on from there. Now, for calculations in this experiment, you may recall being taught CV equals CV, which is dilution calculation. And then you might have been taught MV equals MV because of molarity, but basically you can use any concentration term and use that for dilution. So if I was trying to go from 0.5 molarity HCl to 0.1 molarity, I can use this equation to calculate it. Now, sometimes people use this equation to do stoichiometry, but that only works when it's a 1-to-1 mole ratio. Okay, so it only works if it's a 1-to-1 mole ratio in a balanced equation. So you really shouldn't use it for stoichiometry calculations. You should only use it for dilutions. However, the term that we're using in this experiment is called normality. In normality, we can use this equation because the mole to mole is basically accounted for within the normality unit. Okay, the equivalence between reactant and acid and base is within that actual concentration of normality. This means that the balance equation is not needed like it is for molarity. And this term, normality, is used and very popular in industry for that reason. You don't have to have a lab tech knowing how to write a balance equation. They can easily do a CV equals CV type calculation. Now, we don't call it CV equals CV when we're dealing with normality. We call it NV equals NV. Normality of your acid times the volume of your acid equals normality of your base times the volume of your base. Okay, so if you know your titrant's normality and you know the volume of the acid you're trying to analyze, you can figure out the volume of the titrant it takes to get to the equivalence point and solve for the normality of the acid of your analyte. Now, what is normality? Normality is the number of equivalent weight of your solute divided by the liter of solution. So it's basically some equivalence per liter. The equivalence, as I said, entails the mole-to-mole stoichiometry aspect of things. Okay, so I don't need the balance equation. It's already accounting for the number of equivalents of the acid and the number of equivalents of the base within that numeric value of that normality. Now, how does that occur? Well, the equivalent weight of an acid is defined as the mass of an acid that will furnish 1 mole of H plus, or that will react with 1 mole of OH minus. So the number of H pluses and OH minuses are within that actual concentration of normality. So it's kind of like having that balance equation within it. Example, HCl. HCl, strong acid, breaks up into H plus and Cl minus. So there's one H plus equivalent in HCl. Okay, one equivalent for every 1 mole of HCl. If I talk about H2SO4, there's 2 H pluses for every H2SO4, so there's 2 equivalents for every mole of H2SO4. If I talk about a base, sodium hydroxide, for every 1 sodium hydroxide, I give up 1 OH minus, so there's 1 equivalent per mole of sodium hydroxide. So those are the numbers that are basically involved when you start balancing equations. That's why we've got to balance and put coefficients. We basically accounting for the number of moles of H plus and no H minus that are basically neutralizing each other. Calcium hydroxide has 2 moles of OH minus produced for every mole of calcium hydroxide. So that's all involved in that actual calculation of the normality. That's why we don't need the balance equation, and that's why, since we're talking about equivalence per mole, we can use this equation, okay, NV equals MV. And that's basically all we're doing in this experiment, is using this equation right here, NV equals MV. And as I said, we can use it because it's normality. We can't do it in molarity. Molarity is not, you shouldn't use molarity for CV equals CV. The only way that works is 1-to-1 mole ratio. Everything's not 1-to-1. Okay, most of the time it's not 1-to-1. Now, acid-base equivalence is real easy. When you get to redox equivalence, it's a little bit more detailed because you got to write the half reaction to figure out how many electrons are being transferred. But we'll talk about that when we get to experiment 16. All you need to know in this experiment is NV equals NV. Alright, what's the goal of this experiment? The goal of this experiment is we want the average normality plus or minus the standard deviation of the sodium hydroxide when we're standardizing it. And we also want that for an unknown acid. So we want the average normality plus or minus standard deviation for the standard. Okay, that's our sodium hydroxide. So we're going to run an experiment. We want to do a minimum of 3. Okay, we want to get precision. We in control of precision here. We want 3 good runs. So you should do it till you get plus or minus 0.05 normality. That's around plus or minus 0.5 milliliters. Okay, if you have results that are all over the place, then you got to do more. You want to do so you get at least 3 before you call something a standard solution to at least plus or minus 0.05 normality. That's going 1 less decimal place than we normally do if you're doing an analytical experiment. Typically you want to go to 0.005 normality. So giving you a little bit more flexibility here. And then you also, once you get that standardized, then you do it for the unknown. You do the exact same experiment, except now you know the concentration of your sodium hydroxide. It's still your titrant. You put an unknown acid as your analyte, and then you're trying to figure out that concentration of the unknown. Once again, you want to do that till you get 3 runs that are very close in what you think is the normality of that unknown acid, which should be around 0.5 milliliters runs if you want to get it that close. Now, significant figures, you're going to do your normality, which is going to be based on the original normality on that standard solution and your calculations of sig figs. However, for the standard deviation, what do you use for this number of sig figs for that? Well, your sig figs and your standard deviation should match that of your average normality. It is your measured value. You can't be more accurate in your error analysis, which is what a standard deviation is, than in your measured value. So say you come up with an average normality of 0.123. Now I want to do my standard deviation, and I come up with that, and I do my calculation. I get 0.00215. Where do I stop my sig figs? Well, my measured value is only to the thousandths place on this particular number. Therefore, my standard deviation, I can only be as accurate on my error as I am on my measured value. It has to stop at my thousandth place as well. So I would report this number as 0.123 plus or minus 0.002 normality. Realize that that standard deviation has a unit as well, so typically if you write it this way, you just put the normality at the end. That's 0.123 normality plus or minus 0.002 normality. Here's another example. 0.12, this is only to the hundredths place, and I measured my standard deviation to 0.00567. So once again here, I need to match them. This is to the hundredths place, so therefore my Standard deviation cannot be any more than to the hundredths place. Now, it can be less. I can have less accuracy on my error analysis than I can my measured, but I can't have more accuracy on my error analysis than my measured value. So this I would round off and make it as 0.12 plus or minus 0.01 normality. Now, how do you calculate the standard deviation? I want the standard deviation. To calculate the standard deviation, that is the summation of your data point being your normality minus your average normality. That is squared, so you do that for your first run. So you take your first one minus your average, square that, add your second run minus the average, square that, etc. Okay, so what you're looking at here is so you're taking the square root of, you're talking about your data set 1, subtract your average, that is squared, plus your data set 2, data point, minus your average, that's squared, then add your third one, squared. Okay, so if you got 3 different values of normality for that standardization or your unknown, you add those up, okay, subtract them from the average and squared it, divide that by n, in this case it's 3, minus 1, n being your number of experiments, okay, number of data points. And the whole thing, you take the square root of the whole thing, and that'll give me my standard deviation. Now, if you did 4 runs, you did 5, whatever that you're going to put into it, then you would have 2 more sets of data, x4 minus x bar squared, etc., and it'd be divided by 5 minus 1. In this experiment, we want at least 3 good ones. Okay, if you have a bad one, a 4th one that's really off, okay, we just throw that out. We're trying to standardize this. We just want the ones that are close. Okay, you probably could have had an error with an air bubble or something else on that one that's really off. So you're trying to get precise data to do your standardization. For your unknown, well, you're just trying to get good, precise data to be able to turn in the results. In this experiment, one change in this experiment, they tell you to use 20 milliliters in the manual for your acid quantities. We're going to just use 10. We're only using 10 milliliters because we want to make sure that we have our experiment end in 1 burette reading. So 10 milliliters per run, we take a pipette and pipette 10 milliliters into the Erlenmeyer flask to do the run. How much data, chemicals do you need to run the experiment? You need 150 milliliters of the 0.1 normality of sodium hydroxide. You need 45 milliliters of the 0.1 normality of the H2SO4 standard solution. Notice whatever the value is on that bottle is the concentration of that standard solution. So whatever it says on that bottle, we record that. That is that number for that H2SO4 that we'll be using in this experiment. You also need 45 milliliters of unknown that will be assigned by the TA. That should be plenty enough to do 4 runs of sodium hydroxide and 4 runs of your unknown. The manual says something about doing 2 runs and throwing out the first one. We don't throw out anything. I want you to do all of them. You want 3 good runs and you average 3 runs. Okay, you do not throw out a run unless it's far off from your normality of the other ones. Okay, if it's not in that plus or minus 0.05 normality. Okay, so you want 3 runs you average. Okay, and I want a standard deviation for the 3 runs. Anytime you average at least 3 things, you should be a standard deviation. As you're doing this experiment, make sure that you're writing the procedure in your notebook as you do it. You don't write the procedure later. You don't write the procedure when you go back to the classroom across the hall. The procedure is written as you do it in the laboratory, in your notebook. Everything is written in the notebook, not loosely. Okay, not in the lab manual either. All data is recorded in the notebook as you do the experiment. I want to be able to have a procedure enough that you could reproduce this experiment if you wanted to hand it to somebody else to do again. Now, when you do the second run, you can just say, procedure done just as well as the first experiment, except X grams. It was a different mass of something or different size sample or whatever. But basically somebody needs to be able to reproduce this experiment from your notebook. Okay, for our lab reports, we need to write discussions. So let's give you an example of what I'm looking for in discussion. Okay, discussion is taking the data, analyzing it, using the theory that you've been talked to about, and basically putting it in a format that's understandable and readable. Here's an example. Here's an example of miscibility, solubility, and a density experiment. We start off the discussion. This experiment investigated the concept of miscibility, solubility, and density. One liquid is considered miscible in another when they form a homogeneous solution. This occurred when dichloromethane and hexane were mixed. This is understandable because both of these substances are nonpolar in nature, and only substances with similar polarity tend to be miscible. This explains why water, which is polar, was immiscible in the other two substances. Basically, what I'm looking for from you is to define the theory. What theory are we talking about in an experiment? Then give an example of it from your experiment, and then use that theory to explain the results. And that's what's happened here. First off, we talked about the theory, then we gave some results, and then we used that theory to explain the results. Okay, that's basically what we're looking for in the discussion. Okay, taking what we discussed in our lecture, what you got knowledge from your textbook, whatever, the theory behind that experiment, what did we find in our results, and how they're related. Here's another example. Solubility is a term used when a solid or gas dissolves in a liquid solvent. The solid iodine crystals were soluble in both nonpolar substances, as evident by the changing color of solution from colorless to purple. The iodine was not soluble in the polar substance of water, which did not change color. Iodine itself is nonpolar, so once again the results are expected. Once again, talks about the theory, talks about the results, how are the two related. Another example. The density of each substance measured was determined to be 1.30 grams per milliliter for dichloromethane, 0.677 grams per milliliter for hexane, 0.977 grams per milliliter for water. All these results are less than 3% from their respective theoretical values, indicating good accuracy. Precision cannot be analyzed due to the fact that the experiment was performed only once. Another thing that you need to talk about in your experiment is precision and accuracy. When you talk about precision, that's how close an agreement the different runs are from number 1 to number 2 and number 3. Precision. That tells you nothing about accuracy. You can be very precise. I'll have the exact same number for every run and be wrong. Okay? That just tells you how good an agreement your results are. That comes from your standard deviation. The smaller that number is, the better your precision is. In this particular example, they only did one run of everything, so they couldn't talk about precision. Okay? However, they could talk about accuracy. Accuracy is how close you are to the actual value. So they did a relationship between what they determined and what the actual theoretical value is and came up with some percent error. Okay? And talked about the accuracy. Most of your experiments in this semester, you can talk about precision. You will not be able to talk about accuracy. You will do 3 runs or more, so therefore you can talk about how precise your data is, but you don't know what the final answer really is, so you can't talk about accuracy. So make sure you know the difference between the terms and when to use the terms in your discussion. Last thing that I expect in a lab report is to talk about error. Error is not just saying human error, or we could have made mistakes along the way. You have to think about the experiment. I want you to think about what mistakes could have happened, what could be causing problems. Not that I'm saying that happened or didn't happen. What can affect the experiment? I want you to think about it. Here in this example, the main source of error in this experiment was the temperature of the solutions. The theoretical density of the dichloromethane and hexane were not at the temperature of the experimental solution. The density from the CRC was 15 degrees C for dichloromethane and 20 degrees C for hexane. So they did the experiment at room temperature, was not the temperature of the density they were comparing to for their percent accuracy. That's a source of error in their results. I want you to think about the error. Give you more detail on how to think about error. We're going to talk about that in the next example I have. Enthalpy experiment. Here's a discussion. This is where we're going to talk about the theory, what we found, and how it fits in. When reactions occur, there is an accompanying change in energy. Typically, this energy change is in the form of heat, which is called enthalpy. Reaction usually either absorbs heat, endothermic, or releases heat, exothermic, to the surroundings. The enthalpy of the reaction between HCl and sodium hydroxide was investigated. It was found that a negative 58.3 kilojoules per mole of heat was involved in this reaction. This means that when HCl and sodium hydroxide are reacted, 58.3 kilojoules per mole of heat is released to the surroundings, indicating that this reaction is exothermic. The surroundings in this experiment included the salt solution produced in the reaction in the coffee cup calorimeter. So once again, theory, results, how it's related. Also notice in these two examples, we did not say anything about we did this or I did that. There's no first person in these. It's all written in the third person. Okay, we do not use I, we, anything like that. The sample is weighed. Okay, those type of things is what we say. This experiment was conducted 3 times with good precision, standard deviation of 0.1 kilojoules per mole, and an accuracy of approximately 5% from the theoretical enthalpy of HCl. So in this case, we did several runs, so we can talk about precision. You take that standard deviation to see how good that is. And then we can talk about accuracy because we know what the actual enthalpy of HCl is, and we can do a little error analysis on that. Okay, so you talk about theory. Okay, top, we're talking about theory, what we find, how it's related, precision / accuracy. Then lastly, you need to talk about error. There are 2 main sources of error that could interfere with the accuracy and precision of this experiment. First, the calorimeter used in this experiment was a styrofoam cup with cardboard being placed on top of the cup as a lid. Heat could escape through the unsealed lid during the experiment, causing inaccurate temperature readings during the experiment. Second, the 2 thermometers used in the experiment were not calibrated, one each in the HCl and sodium hydroxide solutions before they were mixed. The 2 thermometers indicated different room temperatures. When the 2 solutions were mixed, the same thermometer that was in the HCl solution was used in the mixture, making the measured change in temperature for the sodium hydroxide solution questionable. However, reasonable results were still obtained despite using the calorimeter that was non-technical coffee cup with cardboard. Just because they tell you how to do it in a procedure and say do something, it doesn't mean that it's not instilling some error. Okay, I want you to think about the experiment and talk about those things that could be happening. They noticed in this experiment that we used 2 thermometers, then we mixed the solutions together and used 1 thermometer. Well, both of them didn't read the same temperature at room temperature, so that's going to instill some error there. Other examples if I tell you to pipette 20 milliliters and I give you a 10 milliliter pipette, well, you got to read an initial, you got to read a final both times, so you instill in double the error. Okay, you should get a 20 milliliter pipette if you want to cut the error down. Okay, those type of things. Think about the experiment of what can be affecting it. Okay, I don't want the normal human error or this or things went well. I want you to think about the experiment and give me some details about how different things affected the results of that experiment. So, to give you some hints about today's experiment, okay, these are questions you should be trying to answer. Okay, what are we doing and how did we demonstrate it? That's what I'm trying to figure out. What is the theory behind this? What are we talking about in this experiment? What are we doing and how do we demonstrate it? There were terms we discussed in the lecture. Anything I give you in a lecture, you should be trying to put that back in the discussion somehow that's somehow related to the experiment. Okay, there were terms we discussed in lectures such as standardization, equivalent point, endpoint. How are they related? How did you see those in this experiment? Why do we do standardization, and how is it related to today's experiment? Why did we do the sodium hydroxide experiment? Why can't I just calculate sodium hydroxide and use that value and then do my unknown? Why I had to do standardization first and then do the unknown? Okay, why did we have to do sodium hydroxide then do the unknown as? What is the purpose of the indicator, and why does it change color? What's going on there? Why did I have to put that indicator, and why did I stop when it turned pink? These are the type of questions you need to be answering, okay, when you're trying to do your discussion. I'm helping you out on the first one, but on the rest of them, you're on your own. Okay, you're going to have to figure out, pull this together to write your discussions. I expect a nice, good, detailed discussion. I'm not looking for quantity, I'm looking for quality. Okay, make sure it's quality and gives me information about why we're doing it and how it's related. You basically must convey that you understand the experiment to me. That's the bottom line. In that discussion, you need to convey to me that you understand what you did and you just didn't get some results and put some numbers together. Okay, I want you to give me details that tell me that you understand the experiment. The last thing that we have to do for this experiment, after you did your lab report and you did your discussion and all that, on the last page, one page over, okay, add one more page to your lab report. I want you to answer an additional question. This is totally separate from the rest of the lab report. Okay. During general chemistry, several acid-based concepts have been discussed. After your conclusion in Experiment 13's lab report, name 3 acid-base concepts that describe the acid and base behavior of molecules and ions. In your textbook, back in Gen Chem 1, you learned about acid-base theory. Okay, some concepts. I want you to name them. Okay, name the 3 acid-base concepts. Then for 2 of those concepts, I want you to explain the definition, give me the definition of 2 of them, then give a reaction and indicate the acid and base component. Okay, so give me a reaction and show me which part is the acid, which part is the base, based on that theory. Okay, so name the 3 theories, 2 of them, define the two of the theories, then give me a reaction and show me what's the acid and what's the base component by that definition.